various dimensions to that. And you try and classify those. And if a second question comes up, there may be a predictive component. So 13 seconds, any questions?
How do you know the local or local? Like, is it kind of range? Because with Alzheimer's, it's so wrong that sometimes people make the only features because you only know that somebody has Alzheimer's.
So in this case, these are the training data. So you actually have all the known results. And then we have test data against which you try and run your algorithm.
But like for this kind of question, is that go to Arno, we're going to have like 3 full days to discuss all the details of like why people, normal people are normal. It's like a really good question. I'm also very curious. Okay.
You okay, shut your down. We can't start it until this room. Okay. Actually, let me give this to somebody else. I will help with you. Yeah. There, you're good. You're good.
All right. So I'm Andy. I present a really small project. It's about deep brain simulation, which is a highly efficacious treatment option for Parkinson's disease and other movement disorders, where people actually get electrodes even through the brain. And usually what happens is that these electrodes have like 4 contacts, and people in the hospital don't know which one to stimulate somehow, so they just try out. And this has a really long, leads to a really long hospitalization, and also it's a bit fuzzy process. So to improve this, I wrote a toolbox which can identify the electrodes, which you can see here, based on post-op MRI or CT data, and then visualize it with atlas data. Then you can even set a stimulation volume with, like, say, you want to stimulate conflict number 2 with 5 volts, and then you see how much is stimulated, and you can even implement fiber tracking results with that and see which other regions in the brain will be stimulated with that. So it's somehow simulating the stimulation. And for now, this only works on atlas data. And there's something about the project. It's, yeah, I said all that. So now it only works with atlas data. What I want to try to do here is to segment the supralimbic nucleus, which is the main target area for deep brain simulation, on the patient data, and then visualize it like the patient-specific atlas data in MRI space.
So you need people to help you do the segmentation?
Yes, I have not too much ideas of how to do it, but I have brought the SBM book and some ideas to try out. And I'm not even sure if there's any interest here, but if there is, then just you can help me. Thanks.
I'm interested. Thanks.
Okay, hi, I'm Andrew. I don't have any slides. I'm just going to talk about this. About a software that I've been developing over the last better half of a decade. Originally, it was a Java-based software just to do visualization, 3D and 2D slices. But now I'm developing it more into a general, I guess the best analogy would be an IDE for brain research. So I have implemented database connectivity and project organization. So basically taking any kind of arbitrary project and organizing it in a standard way, and then being able to apply it to pipelines. So this is an open source Java project. It's got a really extensive API, and it's available online for everyone. What I'd like to do is just sort of feel people out here, if anyone is interested in either using it, having me develop with them, or developing it on their own, or just using it as an end user. All of these ideas are open. Yeah, so I don't have slides to put up at the moment, but I do have a lot of visuals to show you if you want to approach anytime during this hackathon. IDE, so integrated development environment like Eclipse or Net. Yeah. called model GUI. So basically, the idea is it's a GUI environment that basically should be able to handle all of these, basically everything. So pipelines and the data sharing and all of it. So I'd like to basically make some kind of integrated environment that will handle all the things that we want. So I'd like you to approach me from all of these areas.
All right, so hello everyone. My name is Samir Das. I'm from the Montreal Neurological Institute. I've dedicated the last 12 years of my life to LORIS, which is a database system for really brain analytics. I'll try to do this in less than 2 minutes, but we'll see if we can. This is what it is in a nutshell. Basically, it's a system that allows you to do data management, acquisition, quality control, basically everything under the sun in terms of big data and collecting it. We have all kinds of other tools that we use with LORIS. So it actually started with a project called the Pediatric Project. We were a data coordinating center. 15 years ago, a colleague of mine, Dario, said, hey guys, there's this thing called the internet. Why don't you use that instead of shipping CDs all around the place? So this was how LORIS was born, and the rest of my life began. It's proliferated all over the world, and I'm running around all over the place, installing it, and so on and so forth. There's a few people here that are actually working on it. One of the projects that's housed in it is Big Brain that's got some buzz last year, so some of you might have heard of it. There's a whole bunch of other multi-center studies that deal with heterogeneous data, imaging, genetics. Oh, I still have 45 seconds. That's great. So if you're interested in big data or large data analytics for the brain specifically, then come. There's a few of us here. Just a quick plug to other software and tools that we're actually using within LORIS. So one of them is Brain Browser. Tariq Sharif, a very great coder, is working on this. He'll give you this pitch after this. So there's a whole bunch of different tools. If you're interested in working with us, then come find me. I think that's all I can do in 2 minutes. Thanks.
Does anybody not have a so
Alright. So okay, so I have 3 comments. So I should have 6 minutes.
no, because he gets half an hour. That doesn't go to other emails.
No. Okay. Alright, cool. So first one is quality control. So recently we started working with big data sets, and you probably know about like processing pipelines, which makes everything automated and great, but you forget to look at the data. So we decided to kind of change that and look at our data. And we started doing those fancy reports that people collaborate on, and we use some statistics here, and then people like put comments on it and all that. And every subject is generally short, and you can see the mean PI, the overlay of the mask, the PSNR map, and then so how the PSNR look in comparison to everyone else in the sample, with the sample is an outlier, you have the registration quality, the motion parameters, so on and so forth. And I put them through reports from generating those reports. And I'm very happy to work on it during this hackathon to add more metrics and more visualization that can help with quality control of data. So this is project number 1. And project number 2 is project number 2 is related to my very favorite neural. Did I hear something? So, neural is an attempt to collect unthreshold statistical mass to improve
simulations and meta-analysis, and hopefully one day we will platform for university. The idea is that whenever we publish a paper or about to publish a paper, instead of just putting coordinates of your statistically significant activations, we put the whole map, and then we are able later on to find among our obviously underpowered studies under threshold but consistent across studies effect. And no, I got 6 minutes. So the idea is that very easy to use and our incentives are well, okay, so this is number 2. I'm getting thrown away.
Ooh, get off the stage.
I have to show you incentive is visualization, and we just got this brand new fantastic visualization from Jack Gallant group by Jake Dow and Alex Cook, which has this really fuzzy cool brains. It's pretty cool, yeah. Okay, and the first project which I'm just going to talk about is called effect sizes. So all this meta-analysis is a bit complicated when all of our statistics are very highly dependent on our sample sizes. And now when we are scanning people longer and we are having more and more subjects, we need to start not only looking at p-values, but start looking at actual effect sizes. Or in other words, more kind of machine learning approaches. With what accuracy our findings can predict the effect. For example, into group, schizophrenics, and health controls. And the project would consist of trying to apply well-known statistical methods to extract effect sizes maps from outputs of commonly software like SPN and FSL and others. Yes, that's it. Thank you. Don't throw me out.
So the first one is it's brainspell. So brainspell is kind of the other end in terms of accuracy to basic projects. So here the data is just the peak locations, but for a lot of papers instead of few papers with a very nice resolution. We have here 9,000 papers, more than 9,000 papers extracted by TAL. Based on that only peak data, and for long there are these papers which you can get keywords for them, so you can so when you query the 9,000 papers, you get a list of the papers associated with your query, and also a volume showing the regions in those papers where it's a more complicated, and you also get the volume showing the utilization of those locations. But then the idea would be, so what I would like to ask for all of you is to help me annotate these papers. So if I were to annotate 9,000 papers, I would be doing that for the rest of the night. But if we do have 5 papers per day each, or 1 paper per day each, while at brainhack we would be able to have a lot of those. So how does the annotation look like? You click on 1 paper, you get the abstract. Normally, the idea is that you go and do the full text. Like you have your analysis from your university, and if you don't, I could help you out with that. So you have here the different tables in the paper. There was only 1. And what I'm asking you to help me to do is to go look in the papers where the table is located, and then annotate it using different technologies. So even if you look at part of the only those are the mesh descriptors, the descriptors put by Content. So for example, if you like this one, you go here, and then you agree or disagree, you will have to open a login, but everything is encrypted on the and then also tell me if the stereotype space is MNI or Calera, and approximately how many subjects were in it. Okay, and then for the annotation, I'm using 3 ontologies, 2 from the cognitive data, 1 for the behavioral domain, and another 1 for tasks. So
So here you can, let's say you are looking for a working memory thing, and then you can just put there and you can be able to say,
Roberto Malanços.
second.
So that's awesome.
But
Like you can come to him.
yeah, so this is just a framework to transform, to represent the brain instead of representing it as an image as we often do, to represent that as a sound. And for the moment, it's just kind of one way of doing it, and if you want to kind of help developing it or using it, you are welcome. So this will be the sound of a sphere, which is not very interesting. Then here's the sound of a small primary. What you are listening is this brain is slicing the brain, and so this slice produces a shape, and I unfold that shape and use it as a waveform that you're listening. So if the brain is more folded, like a human brain, you have high frequency harmonics. The idea is that with your ears you can understand things that you wouldn't be able to understand with your eyes, and maybe there's a way to present the brain data as a sound that will tell you more than
Every room has a piano, so every room has a piano.
yes.
So I'm sure you can. Yeah.
How do you call the project, Roberto?
Brain stem, aura brain.
Oh.
So the new field that aura.
All right. Hello, everyone. So I'm here just kind of bringing my project, Brain Browser, which is a sort of a WebGL and HTML5 like library for visualization. So if I'm going to add 3 words to my description to myself before, it would be not a neurologist. So one of my issues is that I'm like the web graphics guy, and I kind of want people who know brains to look at what I'm working on, make sure that it works with what they're doing. If I can give a bit of a pitch, it's like WebGL now, like now that's accepted, it's being supported on iPads and everything. It's basically like the most portable graphics platform out there. So if you get your stuff visualized in this way, you can look at it on the tablets, on the phones, on the web, pretty much on any computer out there. So to go through what's available, so it's a little bit less. But so you have surface view, which lets you see 3D surfaces. You can apply like set an intensity threshold, apply your intensity. This is a coordinate thickness map that lets you just visualize your data in that way. You can handle UTI track data as well. Track data. There's also volumetric data. There's the volume view. Data in that way as well. Basically, what it is, that's what it does. Under the hood, it's a JavaScript library to essentially insert into a web page, and it'll create that viewing area that you can use to visualize data. And it's like, you start with your rendering, you load whatever model you want, you can apply colors to it. Okay, last thing, and it's open source, so you can check out the repo on GitHub as well.
So maybe last but not least, since there's no one else in line, lots more. So I just wanted to announce that NITRIC, the Neuro-Imaging Informatics Tools and Resources Clearinghouse, here in force, and amongst their many deliverables include the NITRIC computational environment, which is a virtual machine, web-based and cloud-based Amazon opportunity. We like to work with teams to help use that platform as best as possible for you. We also have the image repository and lots of new space that the NIH is making available for sharing data everywhere. So while we don't have a specific project, we are here to help everyone with their projects, and we will make a very valuable reward award to the best use of the NITRIC computational environment and the best use of additional data sharing. That award will be prominent advertising on the NITRIC site itself. So we encourage everyone to take advantage of Nina, myself, and Christian, and help us and help you with these types of environments to support you. And so with my 58 seconds, I'll repeat that for the next question.
Thank you.
We just have to cut for 58 seconds.
There's no rollover minutes in this class.
Dan, do you want to go?
Yeah, but I don't need that.
You just took it back.
Has anybody heard a good joke? Any jokes? Jokes? Okay. Ben. Oh, we're on.
Hello, I'm Paolo. I'm part of the Google Summer of Code, and I'm a web developer. And in Google Summer of Code, Roman and Satya are my mentors. And we are working on Neurostars. Neurostars is a question and answer website similar to Stack Overflow, but related to neuroscience. So people there, they can start discussions and start questions and giving answers, exactly like Stack Overflow. But so why didn't we use the Stack Overflow? The reason is that in order to create a website with Stack Overflow, you need to meet some requirements, and plus you are you want to develop new features that is not possible to be in Stack Overflow. For instance, we want to introduce the feature to share data set, so experiment results using the current protocol, so people can somehow share it together with the neuroscience community. And we want also to have the possibility to integrate our website in other websites, such as you are discussing in a forum or wherever with your mates about something, and there can be a button which says, okay, post this question or start a new discussion on Neurostars, and that's what it's going to be. The code base was initially developed by professors from the informatics community, and there's a big website named neurostars.org, which is very popular in the bioinformatics community, and we share the same code base, and yeah, actually we just started up. And we would like to hear from your feedback, and if you want, you can use it here.
So no slides. My name is Tyler Arconi. So I built the NERSYN for some you've seen. So most of the NERSYN various components are open source on GitHub. The website has not been until now, and it's also built in Ruby on Rails, which is great if you like Rails, but nobody in this community or very few people use Ruby. We're by and large, I think, Python fans. So the goal, well,
I have 2 minutes.
So the goal, one goal is to port the website to Flash and release it so that people can contribute. And actually, there's a lot of other things that I think could integrate nicely. So, for example, I would like to switch away from the viewer I have, which I wrote, to something better. And there's a bunch of candidates. So I think if we can so if people have ideas they want to plug into NERSYN, and you often get emails like, hey, wouldn't it be great if you add this? And the answer is yes, and hopefully you can add it yourself, and then develop more quickly. So if you have any interest, I'll contribute to that. Come and talk to me.
So, you're not going to use the same visualization and expect maybe?
I'm going to talk to Roberto about stealing some of those for sure. Yeah.
Hi, Roberto. I was asking whether for the visualization part, you would maybe team up with you for.
So I'm going to talk about a couple projects. I'm going to start by putting up plug, I guess, for Amazon Web Services, and then talk about another project. So this is actually for all of you. So one of the sponsors that we have for the event is Amazon Web Service, AWS, and they're offering up $100 worth of credits for everyone here. So if you go to this link, you can go and get a coupon code to be able to go and plug into your Amazon account and be able to either use their compute clusters or storage, and that'll be good for a year or so. And so $100 can actually get you quite a bit of computation and also get a chance for you to familiar yourself with cloud computing. And also, this ties in well with some of the things that Dave was talking about with Bitrick, where they actually have a machine image that's already pre-configured with a whole bunch of different neuroimaging software. Then you can more or less like click a button and have that environment up and running and ready to go. And there are a bunch of other little tools you can learn about, things like Star Cluster for running, workflows with SendGrid Engine and other things. So if you're interested in that, come talk to me, and we can get you started with your credits, and you can learn a little bit about cloud computing. And so the other one I got is, all right, and I need stuff. So one of the okay, so this is actually going to start off a cluster of different presentations. So what this is it's an effort to work on data sharing within neuroimaging. So this is something that's out of the INCF, so it's an International Neuroinformatics Coordinating Facility. And we have a working group called the Neuroimaging Data Sharing Working Group, or NIDASH, and we've been working on standards for representing different types of information across a variety of different data types. So this is acquisition and raw data, the workflows like NIPIPE workflows, derived data, and also archival services. And so each of these things have more or less like a couple specifications defining exactly how you can share that type of information. And so we're doing this in a collaborative effort. Is that the signal?
No, that's just nervousness.
So I'm not going to talk about this a ton. We have a few other people that are going to help and talk about specific pieces. But one of the things that I'm interested in working on is representing more or less like raw data or neuroimaging resources like Loris or XNet and others, where we'll try to come up with a representation that you can query and develop apps off of. And so anybody that's developing apps, working with IPython notebooks, those kinds of things, we're going to have representations of neuroimaging resources that can be queried.
No, perhaps one thing you want to add is that this is an interoperability standard. It's not intended to replace an existing database, but to bring databases to work together.
Exactly. So this is more of it's a collaboration for how do you take all these different resources and start coming up with ways where they can interoperate.
And we'll be sitting in the room, which is going to be just next to the on the right at the exit of the small room, so at the top of the stair. So if you want to sort of hang out with the data sharing guys, that's where we're going to be. And guys is both of you.
Yeah, there you go.
And also, Michael Riemann will come as well. He's part of the group, and we've come. And we are going to have a unconference on Friday. I don't know whether it's Friday. I think Friday afternoon in this room. So that's yeah.
So my name is Tom Nichols, and one of the projects is called OHBM apps, and it's basically trying to use what we have done. So far, what we focused on is there's some development on describing this end, but the thing that's at this point most mature is on NIBM results. So that's trying to capture the results in a formalized form. And the thing that got me going about this is that if you think about any typical fMRI study, there's hundreds, maybe 1,000 hours that go into generating that data. You know what happens to that data at the very end? someone looks at their SPM output and it really or maybe they copy and paste it from their tabular output into their table. Isn't that insane? I mean, they spend thousands of dollars or pounds, and then there's like a copy and paste at the end. And your neuroimaging data analysis software knows virtually everything about your study. So why does that have to be lost? And so the whole point of NIDM results specifically is to capture that way in a standardized format that's common across FSL, SPM, ACME, whatever, and then be able to share that and be able to do cool things with it. So that's the focus of NIDM apps, is to try to come up with ideas like, okay, now that we have this data and we have a number of data sets represented in this format, then let's create these tables automatically in some machine-generated format. Why not create a machine-generated results methods section? Your software knows everything you did. Why not have a prose version of that? Since it's been now also represented in this format, could you convert this tool? Some tools do like to do the coordinate-based meta-analysis. Could you convert this information to the format that is accepted by the meta-analysis tools, ginger ale or APA, or just other things? So come and stop by and talk to us.
Just the dust that maybe.
Any other talks without a computer? So one day, setting up, I'm going to do my pitch. So I'm going to interact with many of the people I've heard here. I've tried to make a concerted effort not to be take the lead on a project, to be involved with the project. But still, there's this thing I'm going to kind of work on, hopefully with Tariq. So I really like quadruptors because I think they're really nice things. And I've heard about like tongue stimulators. So blind people, you can like have a camera and stimulate what the camera sees on their tongue electrically, and they can actually perceive their environment, avoid obstacles, and navigate, because the tongue is like, if you see the homunculus, you have a lot of sensitivity here. So I thought the two things were awesome, and I'd like to put them together. So I would basically like to have a quadrupter navigating a difficult environment with sensors telling the distance. Like cats, they have the whiskers, they can feel tight environment. So here you would have captors telling you how far are the different obstacles, and that you get stimulated on your body. So you would have stimulated like up, front, right, left, back. So we're gonna, Roberto suggested to use a swimsuit with like small motors from vibrators from phones. So I would like to have those captors, and that you get stimulated with your basically a vibrator. That could be used as sex toy. I don't know. But, that's not the point. So what I want to get done during this brainhack is to actually have a video game incarnation of the quadrupter flying in the small corridor, and Tariq's doing games and 3D. So I guess now you're going to do a game in 3D. And if that all goes well, we use like the numerical sensors to build a prototype with engineers down the road. But that is very early. But if you have ideas about how to stimulate or better things in the swimsuit, you're more than welcome to pitch in, because I like electric stimulation would be cool. I'm just afraid of like ethics and things like that if somebody gets electrocuted. But
Oh, get up!
So Samir already talked about Boris, which is a web-based neuroimaging platform for databasing, and I work on a very specific subset of it called the data query tool, which is used for querying data and extracting from the database. It's written as a HTML5 JavaScript application, entirely client-side. It uses also many sounding technologies. I just want to do a quick demo of it and before going on. So basically, you have a study, you import your data into it, you select the fields you want to be studying from your whatever instance you're working on. You have various types of data. I'm just randomly choosing some here. You can choose your filters that you want to query on, and then run a query across your entire data set to get your results for whatever you're happen to be looking at. and it could be this nice table. You can extract it through CSV. If that had selected any imaging data, you can download it. You can download the package you could create for. And then there's basic statistical analysis visualization tools here, which you use. It gives you, basic stats on the population that you queried within your study. Gives you, min, max, and statistical terms. It can do some. I don't have any numerical data, so I can give you a histogram of your columns, what you've done. You can do scatter plots of your numerical continuous data and group them by various things that you report in your populations with the data that you ran. I've not selected enough data to do that, but it could generate a pretty scatter plot there. And I'll go to find for people who's working on is this is written. It is written for those, but it's written in a way that can generically work with any kind of data. So if you have data that you'd like to get into this tool, so you can use it for analyzing your data in a very basic way, I can help you do that. And I would like to get some feedback or help implementing any sort of visualization things that would be useful to put into this so that you could access your data on the web, visualize it, whether it's creating a brain browser so you can see your camera directly here together, or other any other sort of visualization or statistical tools that would be useful. Because like Karen, I am not a geologist either, so
we didn't catch your name. Oh, hey.
Oh yeah, I've got 11 minutes. We take it 2 minutes. 2 minutes?
All right.
2 minutes.
So I don't have any slides, but given all the things that have been said, there are a couple of things I want to touch base on. There's a lot of JavaScript expertise around in the room, a lot of visualization tools, and one of the goals, again, in the sense of interoperability, was to create libraries that could be reused. So, or to take things out of existing things into reusable components as much as possible. Because many people develop these wholesale platforms for everything. And if we can create the reusable components, they could be adapted for various things. That's me. All right. There's a bunch of things. So JavaScript, we can talk more about the details of things. Tarek and I have been communicating over email. We'll try to figure out ways in which we can extract these things. Okay. Number 2, anybody here who does real-time fMRI? Maybe. If you're interested, we're trying to create an open platform, basically defining a specification that could be implemented across scanners, so that the same kind of software that's used as opposed to a specific software for a specific scanner. That's something missing, but that's a side project. We can talk separately about it. There are a bunch of NiPipe folks over here, and one of the things we want to use the next 3 days is for re-architecting the next generation of NiPipe, so that we can take into account many of the other developments that are going on. FreeSurfer, how many people use FreeSurfer over here? Okay, quite a handful. How many people do edits on FreeSurfer? Oh, a few? Okay. So as most of you who've done FreeSurfer know that edits are often required to create quality outputs. One of the projects is, could we automatize that process? Could we make it such that we don't have to do edits anymore? I see a head going that way. So one of the things we'll do at some breakout session is to sort of brainstorm what's possible, what's not, and figure out if we can actually implement this. And then the final plug I put along the lines of the entire NIDM stuff, we are looking at ways in which we can interoperate. So NIDM being a standard, so we can incorporate it into your software in some ways. Even if it's preliminary, that gives us a start. So let's work on various tools that people are developing and trying to get these things integrated right away. All right.
Are there any other announcements, jokes? Oh, there is an announcement.
I don't really but I'm starting to feel inspired. Some kind of brain art that So if anyone has any ideas, I'll just be like sitting on that.
I just had an idea because when the projects were presented, I thought that real time, like today, we want to analyze data quality, so that's not virtual, we have real time data
Any other thoughts, projects?
So at this stage, we can start to shake up the room a bit, and people can. I think the lunch is going to be available to grab in 20 minutes or so. It's right downstairs next to where we had breakfast. There are these very nice prepared kind of box lunches. You grab one, and you can go and hang out and get started. Start meeting people and join the project and start from groups, and we'll basically have that going for the afternoon. Enjoy.
So do we have a space and where the projects are listed and people can say where they are?
We will the brainhack website, and so you'll find all the links that were up today, as well as all the projects are currently available there.
I put the note of what projects are going on, and people can put where they are and be happy.
Perfect.
There is a Google Doc link, but that went on Neurobureau, so we'll need to figure out.
Getting that over to brainhack. There's some cross. There is there a mailing list is a very good question.
There's the Gitter thing.
There's the Gitter thing.
None of them I have.
Yeah.
I wonder if we could just password feed them, but one email address.
Yes, if you registered on site here today, find me and we'll be sure to get you on the list. We do have a list there, vision.
This is suggesting you just make a little flag so that you can flag this table and say
Right. Yes, I think we can do that.
Yeah, I'm a little confused because it's like this paper, but
yeah, we go site.
Yeah,
Site.
I mean, the workspaces are not that big. There's not that many of us, so just wandering around, you get the feel of who's doing what and who's having to learn. It's all like, hungry, primitive. Okay, so bye bye.